Okay, so welcome to this next video in which we are talking about how to measure intracellular, by which we mean cytoplasmic, calcium levels using Fluo-4. Okay, so we're now going to talk about the star of the show, which is Fluo-4. Okay, right. I am not going to show you the full structure of the Fluo-4 molecule. Instead, what I am going to do is I'm going to draw a box, okay? And I'm going to write Fluo-4 in the box, and then I'm going to have the four important groups, as far as our discussion is concerned, coming off this Fluo-4 molecule. Now, nicely for us, the four important groups are exactly the same. So you have four important groups which are all the same group, okay? And this important group is a carboxylic acid group. Now, you might be expecting me to put an alcohol group here now, but the reality is that under physiological pH, these Fluo-4 molecules have generally lost their protons from that alcohol group. So remember, if I draw a carboxylic acid group here, the reason it's called an acid group at all, a carboxylic acid, is that it fulfills the definition of an acid molecule. Now, what is the definition of an acid molecule? Well, the definition of an acid molecule is a molecule which can donate a proton away into solution. Okay, so if we draw this alcohol group in a little bit more detail still, okay, we have a single covalent bond between this oxygen atom and this hydrogen atom here. Okay, and this single covalent bond comprises two electrons. One electron from the oxygen atom, so I'll draw the electron from the oxygen atom here, and one electron from the hydrogen atom. So let's colour these in. So here, this electron here is from the oxygen atom, okay, and this other electron here, which I'm colouring in vivid purple, this is from the hydrogen atom. Okay, so this single covalent bond has two electrons that are being shared by the oxygen and the hydrogen atom. Okay, one electron from the hydrogen, one from the oxygen. Now, what can happen is the hydrogen the hydrogen nucleus, which just consists of a proton. So let me just remind you that hydrogen atoms, most hydrogen atoms, consist of just a proton at the center, okay, and then an electron orbiting the proton. Okay, so here is the electron in vivid purple here, and here is a proton at the center. Now, of course, there are other isotopes of hydrogen. Okay, this is the most common isotope of hydrogen, known as protium, in which you have no neutrons within the nucleus. Okay, but there are other isotopes where you do have neutrons within the nucleus. So, for instance, deuterium, okay, has a single neutron in the nucleus, and then tritium has two neutrons in the nucleus. But the main isotope of hydrogen is protium. This is the most common. So if we assume that this is a protium hydrogen here, then all that it has, other than the electron which it's put into this single covalent bond here, is this proton, which is its nucleus. Okay, so what can happen is this alcohol group can actually lose this proton. This proton can become detached from this structure here, leaving its electron with the oxygen. Okay, that gives the oxygen a negative charge, and then the proton goes off into solution. Okay, that is why carboxylic acid groups are known as acid groups, because they have the ability to donate protons away into solution. Now, this is what has happened here. You've ended up with the carboxylic acid group losing its proton, okay, at physiological pH, which is around 7.45. Okay, so the proton has gone, it's no longer attached to this oxygen, leaving the oxygen with a negative charge. Okay, so this is the group after you have lost the proton, basically, and interestingly, this is no longer an acid molecule anymore, okay, because it can't donate a proton away into solution anymore. Instead, it's actually a base molecule, because it can receive a proton out of solution, okay, because protons can come and bind to the negative charge of the oxygen. So it's actually known as the conjugate base of the carboxylic acid group. Okay, and this is a running theme for all acid molecules, that once they've donated their protons away, the molecule that you have left is no longer an acid molecule because it's not capable of donating protons away into solution anymore. Instead, it's a base molecule because it's capable of accepting the proton back again, and it's called the conjugate base. Okay, so Fluo-4 molecules then, they have 4 carboxylic acid groups which all, under physiological pH, are in these conjugate base forms, as I'm showing here. Okay, so they've all lost their protons from the alcohol groups, and the oxygen atoms now have these negative charges. Okay, so this is our Fluo-4 molecule here, in orange. Right, so this means that the Fluo-4 molecule has 4 negative charges. Okay, right, so we can already see how this might just interact with calcium, and indeed it is going to interact with calcium. But before I come on to that, I just want to talk about the basic concept of what fluorescence is. Okay, so although I haven't shown you the full molecule of Fluo-4, it's not necessary. Okay, we now just need to explore the concept that Fluo-4 is what's known as a fluoro-4, which I think is someone's idea of a joke somewhere. Okay, a fluoro-4 is a molecule which is capable of fluorescence. Okay, so let me just explain to you what fluorescence actually is. So what does this molecule here, what does fluorescence actually mean? Okay, so the concept of fluorescence is that you have molecules which are known as fluorophores. Okay, so we'll say this is our fluorophore molecule. An example would be Fluo-4 here, which are capable of absorbing photons of a certain frequency. Okay, so this is a photon here coming in. Okay, of a certain frequency. So let's call this nu for frequency, and we'll call this the frequency of excitation, Ex. Okay, so we'll call this the excitation frequency, and of course we know that frequencies can be turned into wavelengths, and wavelengths can be turned into frequencies. So we could call it the excitation wavelength or the excitation frequency. It's a photon of a certain frequency or a certain wavelength, basically. Okay, so it's some form of electromagnetic radiation. This fluorophore, this special molecule here, is capable of absorbing that photon. That will give it some energy, and the molecule is then capable of releasing that energy again. But when it releases it, it will release it again as a photon, but the photon will now have a lower frequency. Okay, so I'll put nu here for frequency, and I'll call it Em for emission. So this means the emission frequency. Okay, and the emission frequency is always lower than the excitation frequency. So this number, nu Em, is absolutely always going to be smaller than nu Ex. Okay, and that's because, remember, the frequency of electromagnetic radiation is intricately connected to the energy of that electromagnetic radiation. Okay, the higher the frequency, the higher the energy. So we're putting in a photon of a high energy, and we're getting out a photon of a lower energy. And of course, that's the only way this really could have happened. You can't put in a photon of low energy and get out a photon of higher energy. Okay, so this is what is meant by fluorescence. It involves molecules known as fluorophores, which are capable of absorbing photons of one frequency and then emitting the energy as a photon of a lower frequency, which therefore has lower energy. Okay, so let's see this in the context of Fluo-4 specifically. So now let's say this is Fluo-4. Now, we're going to go over from using frequencies to using wavelengths, but of course we know that one is just inversely proportional to the other. Okay, so we'll call this the wavelength of excitation now. Okay, and the wavelength of excitation for Fluo-4 is 485 nanometers. Okay, which is actually in the visible portion of the spectrum. Okay, that is blue light. So you can shine blue light onto Fluo-4 molecules, okay, and what then do the Fluo-4 molecules emit? Well, the frequency of emission, the wavelength of emission is 525 nanometers. And you might say, hang on a second, but that's gone up. I thought you said it always went down. No, I said the frequency went down, but remember, frequency and wavelength are inversely proportional. So when frequency goes down, wavelength goes up, okay? So the wavelength of emission is always actually going to be higher than the wavelength of excitation, okay? So the wavelength of emission is 525 nanometers. That's green light. So you can shine blue light on Fluo-4 molecules and get green light coming out again. Okay, that is the property of fluorescence, that you can shine one color electromagnetic radiation, and of course it doesn't have to be in the visible spectrum. We like it when it's in the visible spectrum because then we have a decent understanding of it. It can be in other portions of the electromagnetic spectrum. You shine a photon of one frequency on, get a photon of a different frequency off. That is the principle of fluorescence. Okay, right. So, what's so incredible about Fluo-4? How does this all relate to calcium? So far, all we've seen is that I can shine blue light onto Fluo-4 and get green light back again. Well, you can do that. You can take pure Fluo-4 molecules and you can shine blue light away onto the Fluo-4 molecules, and the amount of green light that you will get back is not great. Okay, so Fluo-4 is not actually that great at doing this. However, if you mix the Fluo-4 molecules with calcium ions, what happens is these negatively charged oxygens here, they bind to a calcium ion. So what will happen basically is one calcium ion comes and binds to the Fluo-4 molecule, and all these 4 oxygen atoms, okay, I'll try and sort of show this, I'll show it on the side here. Okay, so here's a calcium ion, okay, it's come and bound here, and all of these 4 oxygen atoms with the negative charges are going to interact with this calcium ion, okay, like so. So you're getting these 4 bonds forming. Now, when Fluo-4 does this, it becomes absolutely brilliant at fluorescing. Okay, it will now absorb blue light much more readily than before and emit it as green light. Okay, so if you take just Fluo-4 and put it in a solution with no calcium ions in, you can shine blue light away at the Fluo-4 and you will get a bit of green light back again, but it's not brilliant. Whereas if you put calcium into that solution and then shine the blue light on, you will get much greater fluorescence. You'll get loads of green light coming back again. Okay, and the more Fluo-4 molecules that are actually bound to calcium, okay, the more will be the fluorescence that you get. Okay, and that's the genius of it, because what determines how many Fluo-4 molecules are bound to calcium? Well, it's the calcium concentration. So now we have got a way to link calcium concentration to the fluorescence signal. And if you haven't understood that, don't worry, I'm about to explain it again. Okay, so basically, if we have a solution here, okay, like so, and we've got loads of these Fluo-4 molecules here, okay, and let's say initially we have a solution where there are no calcium ions, okay, so initially the fluorescent properties of these Fluo-4 molecules is very bad. So you're shining blue light on, I'm shining loads and loads of blue light onto my flask, but the amount of green light that I'm getting off is, you know, pretty pathetic. Okay, now what I'm going to do is add a little bit of calcium. So I add some calcium in, and some of the Fluo-4 molecules are now going to end up with calcium ions bound to them. Okay, so here is a calcium ion. There will be some free calcium ions, because not all of the calcium ions will bind to the Fluo-4 molecules. Okay, it will be an equilibrium, which we'll come back to later. Okay, so now some calcium ions are bound to some of the Fluo-4 molecules. So now some of the Fluo-4 molecules are better at fluorescing, which means that the fluorescent signal will have gone up, but it hasn't yet got to the maximum it could be. Okay, then if we pour in more calcium ions, then more of these Fluo-4 molecules will end up with calcium bound to them. Okay, shown here in orange. Now the fluorescent properties are going to go up even more, so the amount of fluorescence we'll get is going to increase. And note, we have not changed the amount of blue light we are shining on. The blue light has been kept constant. This is not changing. Okay, but the fluorescent signal is changing simply because we are adding calcium into the solution. Okay, and then, you know, we could take this even further and get more and more Fluo-4 molecules. Eventually, of course, we'll saturate it. We'll get to the point where pretty much all of the Fluo-4 molecules are bound to calcium, and then you can't get any higher. Okay, but the genius thing is that with the concentrations of calcium inside the cytoplasm of cells, you don't saturate your Fluo-4. Okay, your Fluo-4 molecules will not all be bound to calcium with the concentrations that are inside cells, which means that we can look at this. Well, we're in the meaningful portion basically, because, you know, if calcium concentration was too high, okay, all of the Fluo-4 molecules would be bound to calcium all of the time. Okay, and then increasing calcium further wouldn't change the fluorescence. So that doesn't give us any information that would allow us to work out calcium concentration from the fluorescent signal. But if we're in that range of calcium concentrations where not all of the Fluo-4 molecules are bound to calcium, okay, and thereby increasing calcium increases the fluorescent signal because it's increasing the number of Fluo-4 molecules with calcium bound to them, then we can get some meaningful information out of this. We can use the fluorescent signal to infer calcium concentration within the solution, and that's the basic idea. And don't worry if you're a little bit fuzzy with it at the moment, we will see it in much more detail because we now need to try and actually calculate how to, well, work out calcium concentration from a fluorescent signal. Okay, right, but before we discuss how to actually work out what a change in the fluorescent signal actually means with regards to calcium concentration, I firstly want to take this back onto the level of cells, because at the moment we're just dealing with solutions in flasks, okay? I want to take this into cells. How are we going to get this dye molecule into cells? Okay, well that actually does present more of a problem than you would think, okay? Because let's say we have a cell here, okay? So this is a cell. If we've got our Fluo-4 molecule, which at the moment is not bound to calcium, so at the moment we'll assume our Fluo-4 molecule is not bound to calcium, how is this Fluo-4 molecule going to get across the cell membrane? Okay, even if it is bound to calcium, we can still ask the question, how is it going to get across the cell membrane? Okay, you see the problem is that the cell membrane is a layer of phospholipids. It's extremely hydrophobic. This molecule has got 4 negative charges, and even if it binds a calcium ion, it's still got 2 negative charges overall. That is not going to be able to get through a cell membrane. Okay, so we're confronted with this problem that Fluo-4 isn't actually going to get into cells even if we pour it on top of cells. Okay, so what we have to do is modify the Fluo-4 molecule to get it into cells. So what we do is we create another molecule which is known as Fluo-4, and then people put an AM after it. Okay, now what does this mean? This means Fluo-4 acetoxymethyl ester. Okay, so this AM here, what this stands for is acetoxy, so that's the A, and then the M is for methyl. Okay, so the acetoxymethyl ester. So we are going to use the acetoxymethyl ester of Fluo-4 to load cells with this dye. Now, what does acetoxymethyl ester mean? Well, basically, what it should be screaming at you is that we're going to stick ester links onto these 4 carboxylic acid groups, and that's exactly what we're going to do. Now, there's still the technicalities of what does acetoxymethyl actually mean. Well, basically what we're going to do is we're going to take each of these carboxylic acid groups here of the Fluo-4 molecules, and we are going to esterify them, okay, with the acetoxymethyl group. Okay, so let me show you what we're going to add on effectively. Okay, so you can imagine having a molecule like so, where you have an alcohol group with a methylene group here, okay, then bound to an acetyl group here. Okay, and I'll explain how this fits the name in a moment. So, this is what we are going to esterify onto Fluo-4. Now, each of the carboxylic acid groups are Fluo-4. Now, when you form the ester links, what happens is the alcohol group will come off the carboxylic acid group, the hydrogen will come off the alcohol group of this molecule here, and you will then attach the carboxylic acid group's carbon to the oxygen of this alcohol group here. Now, what that will produce is this carbonyl group here will be linked to that oxygen of this molecule here. Then what you'll have is this methylene group here, and then you'll have this ester link onto an acetyl group here. Okay, this is what is meant by an acetoxy-methyl ester. Okay, let me make sense of the name for you then. So, this group that you have here, which consists of a carbonyl group with a methyl group here, that is the acetyl group. Okay, so the aceto here, or at least the acete here, means that we've got this acetyl group. Now, you then have the oxygen in the middle, okay, here in purple. That is the oxy portion of this name. So the acetoxy refers to the acetyl group, and then the oxy portion here. Okay, and then you've got the methyl portion, which is this methylene group here in orange. Okay, so that's the acetoxy-methyl. Okay, and then the whole thing is an ester that's attached onto the carboxylic acid group. So this portion here, this ester link, that refers to the ester there. So, for each of the carboxylic acid groups of the Fluo-4 molecule, shown here, you are going to attach on one of these molecules here to create 4 acetoxy-methyl esters, okay, and that's what is meant by Fluo-4-AM. You no longer have the problem with this molecule having 4 negative charges. Okay, those are completely dealt with now, because these oxygens, you know, they're not even an acid group anymore. They can't donate protons off them. Okay, so that sorts out the problem of this being ionized under physiological pH, basically. Now what you can do is you can mix, well, you can put the Fluo-4 molecules into a solution. Okay, you put the Fluo-4-AM molecules into a solution, and then you bathe your cells in this solution. So generally, when you're doing these sort of assays, what you will be working with is a 96 well plate. Okay, so let me just draw one of these here, like so. We'll just concentrate on one well, but you will generally be doing it for a 96 well plate in some sort of high throughput assay. Okay, so you'll have a plate where it has 96 wells, and I'm not going to manage to draw 96 of them, but you'd have 8 wells along this axis here, and then 12 wells along this axis here, to overall give 8 times 12, that's 96 wells on the entire plate. Now at the base of each of these wells, so if I draw one of these wells here, okay, at the base of each of the wells, down here, you will have cells growing on the base here. Okay, so in green, these are the cells growing on the bottom. What you will do is you will remove whatever is already on the cells, so for instance the growth medium, okay, and then you will add on top of the cells a solution containing the Fluo-4-AM. Okay, now the solution will need to contain loads of other things as well to keep the cells alive. Okay, but we won't go into the technicalities of what the exact constituents of the solution generally are. The point is that you've put on the top of these cells a solution containing the Fluo-4-AM molecules. Okay, right. And now what you're going to do is you're going to incubate the cells with this solution for usually it's about an hour. Okay, it's usually done in darkness because Fluo-4 is actually light sensitive. Okay, so you don't want it to be exposed too much to light, otherwise it could destroy the experiment. Okay, so during this time, during this hour-long incubation period, what will happen is these Fluo-4 AM molecules will be moving around the solution, okay, and some of them will just happen to hit cells. They will go through the cell membrane, because they can now easily get through the cell membrane, and they will go into the cytoplasm of these cells on the base. Now, this is the genius part, because you might think, well, great, but hardly any Fluo-4 AM molecules are actually going to end up down there, but there is a genius part to this, okay, and this is the ingenious bit. Basically, Fluo-4 AM molecules go into cells, but they never come out of them again, okay, and let me explain the reason why. Okay, so in comes the Fluo-4 AM molecule into the cells, okay. Now, inside the cytoplasm of cells, there are enzymes which are capable of breaking ester links. Okay, and these are rather marvelous enzymes. They just break ester links. They don't really care what molecule they break them in, they just break ester links. Okay, they hydrolyze them. Okay, these are known as non-specific esterases. Okay, and all over the place in experimental biology, non-specific esterases are used. Okay, we take advantage of them to trap molecules inside cells. So this non-specific esterase is then going to cut the acetoxy-methyl esters off the Fluo-4 molecules. So it's going to break this bond, return these groups, these 4 groups of the Fluo-4 molecules, back to being carboxylic acid groups, okay, just like this. And now this molecule cannot cross cell membranes, so it is utterly trapped within the cell. Okay, now note, which compartment of the cell is it trapped in? It is trapped within the cytoplasm. The esterases are within the cytoplasm. So Fluo-4 molecules within the cytoplasm are going to get trapped within the cytoplasm. In other compartments of the cells, for instance the endoplasmic reticulum, they're not going to get trapped. Okay, so the Fluo-4 molecules will end up trapped within the cytoplasm of the cell. So it is a cytoplasmic dye. Okay, now as I say, you are going to incubate these cells with this solution containing Fluo-4 AN for generally around an hour. Okay, that's always the time that I've done this for. Okay, and in that time what's going to happen is these Fluo-4 molecules are all, the idea is that they're pretty much all, well, a good number of them are going to end up within the cells. You're going to load these cells full of Fluo-4 AM. Well, not bursting full, obviously, but the idea is that, you know, some will go in and get trapped, but gradually you'll just get more and more going in. So you're going to load a lot of Fluo-4 into the cytoplasm of your cells that are on the base of your well. Okay, then what you'll do is you remove the Fluo-4 containing solution. You put it back in another solution. You generally leave it half an hour just to allow everything to normalize, okay, to allow Fluo-4 AM molecules that have not been deesterified to leave the cells and go back into this solution out here, and allow all the other Fluo-4 AM molecules that are going to be deesterified to be deesterified. Okay, so you allow everything to be deesterified or end up back in this solution, basically. And then what you're going to do is remove this solution, add some fresh solution, and then you're ready to run. You're ready to add your agonist for the receptor, and you're ready to see a change in the fluorescence from the Fluo-4 molecules within the cells on the bottom of this well here. Okay, and we'll go into what's going to happen when we stimulate with our M3 agonist, for instance, in the next video.